Morning, everyone. Morning, Hala. Everyone's asleep. Oh, good morning. I hope everyone's doing well, making it through this semester. Happy Thursday, or as my husband says, Friday Eve. All right, let me share this. So today we're going to do something a little different. We're going to kind of re-examine some of the things that we've already covered in the course I'm just making everything big here on my end. To kind of like put things together and have a, sorry, I'm trying to, all right, well, I'll close this box. Okay. Yeah, it's good now that we have made our way through the non-vascular plants and we've learned a bit about some of the vascular plants. It's good to kind of take a step back and kind of put some of these things we've learned in context together. And we'll also touch today on some of the kind of core course concepts of this class, because it's good to do that. And the core course concepts are kind of like what it's all about, but we have to learn little things along the way in order to kind of understand those core course concepts. So you guys should be able to see my screen, see my PowerPoint screen. Let me know if you cannot. And so today we're kind of going to re-examine this whole idea of like the shift in dominant generations, meaning shift from having a Gametophyte generation be dominant to Sporophyte generation be dominant. So what have we even learned so far in this course, right? Well, from the get-go, we learned that there's a lot of plants, right? Almost 400,000 plants that we know about on Earth. And in the first class, we talked about that the majority of those plants are actually flowering plants, which, oh, Kira, are you having problems hearing me? Maybe leave the Zoom and come back in. Sometimes that solves problems like that if you're having issues like that. Oh. All right, before I go on my rant here, I'll make sure everyone's able to hear me and see things. Okay, yeah, so we've learned there's a lot of plants and we've learned that the majority of those plants are flowering plants, which we haven't even talked about yet, right? We haven't even covered flowering plants yet. But that's a big deal. The fact that the majority of plants on Earth are flowering plants means that having a flower works best as a strategy, right? Because the majority of plants have flowers, so that strategy tends to progress. Oh, okay. You guys are distracting me here. It's not going to work. I'm still going to go through this. Yeah, so like the majority of plants are flowering plants, so that must be a good strategy, right? But there had to be steps. Obviously, we've talked about a bunch of plants so far. None of them have flowers. None of them even make seeds yet. So there must have been evolutionary steps or milestones that led to this whole concept of having a flower. And so one of these key milestones in leading to having a flower is this shift in dominant generations. Another thing that led to this, oh my gosh. All right, hold on. This is crazy. Kiro, you need to mute, hun. I'm trying to mute you on my end and it for some reason won't let me and I don't know why. There we go. Okay. All right, so, okay. It's like, I'm so happy everyone's coming to class today. That's very good, but it's like, come early so I can mute you. All right, milestones. Milestones that led to flowers, right? First important milestone, shift in dominant generations. Going from having Gametophyte generation dominant to having Sporophyte generation dominant. That's super important in the history of plants, but in particular in the evolutionary history of getting to a flower. Number two, increase complexity of the Sporophyte plant. We would never be able to think back to those mosses, think back to even liverworts, and think back to those Sporophyte plants. Those Sporophyte plants were super puny and small, right? Okay, great. I'm glad you could hear now. Those Sporophyte plants and those non-vascular plants were super puny and small, right? Nothing that could ever have a flower on it, for example. So we needed the Sporophyte plant to get more complex, and we need the Gametophyte plant to become more reduced, because ultimately, spoiler alert, ultimately the Gametophyte plant is actually going to be part of the flower. So we can't get there unless the Gametophyte plant gets really reduced. Sporophyte plant, Gametophyte plant, we know what these are, but based on some of your assignments, you don't. So what is a Sporophyte plant? If you don't know this by now, if you don't know this by now, sorry, if you don't know this by now, hon, you are at a disservice to yourself. Okay, a Sporophyte plant is a plant, right? A plant that makes what? Spores. A Gametophyte plant is a plant that makes what? Ultimately, gametes, egg, and sperm, right? Ultimately. Sporophyte plant is diploid. Gametophyte plant is haploid. Sporophyte plant is where meiosis is going to occur, and the result of that will be haploid spores. Gametophyte plant does not need to do meiosis. Meiosis is, you're already dealing with a haploid plant. So on the Gametophyte plant, you're only going to have mitosis and cytokinesis to produce these haploid gametes, right? If you don't know this by now, you gotta step it up. This should be no-brainers at this point. This is our nice summary slide of alternation of generations, right? All plants do this, whether or not you're a moss or a flowering plant. All plants do this. This is the life. You can think of alternation of generations as the life cycle of plants, right? The life cycle of plants. So this is a very good slide to have, but this is what I just said on the previous, right? You have a 2N Sporophyte plant. On that Sporophyte plant are organs. Well, that's not. On that Sporophyte plant are organs, sporangia. Inside of that organ is spore mother cells. Those are going to be the cells that undergo the process of meiosis. If you don't know what meiosis is, you need to go back to that second lecture. And the product of the meiosis will be haploid spores. These haploid spores are going to grow into Gametophyte plants that are haploid. And those Gametophyte plants will grow on them organs that make egg and sperm, right? Gametes. Organs that make egg, sperm, and egg. Sperm and egg will unite. You'll get a 2N zygote cell, which is now diploid, right? Beginning of the new Sporophyte plant. All right. This should be boring at this point. Like we've covered this so many times now. These concepts should start to feel a little bit boring. If they don't, and you're confused, please set up office hours with me. Right. But as I said, every plant on Earth goes through this process because it's the life cycle of plants. But over time, over millions of years, we have these significant evolutionary milestones that lead us to a more complex sporophyte plant and a more reduced gametophyte plant, which are requirements in order to get to a flower eventually, right? All right. So as the sporophyte plants get more complex and as the gametophyte plants get more reduced, we see a switch in the dominant generation. In those older, AKA primitive plants, meaning the non-vascular plants, the gametophyte generation is dominant. Mosses, liverworts, and hornworts. Over time, as we see vascular plants show up, the sporophyte generation is dominant, right? So we've already covered the lycopods and the horsetails as members of those vascular plants. In those members, sporophyte generation is dominant. And as we continue through the rest of the course, every plant we could continue going forward will have sporophyte generation dominant. So why is this? Is how your brain can be thinking, right? Why do we go from this? If we still have mosses, liverworts, and hornworts today on Earth, living right now in Newark, in New York, in Michigan, right? So that system works. So what's the point of evolving to have your sporophyte generation be the dominant generation? What's the point of that? And right, and what kind of led to the need what led to the need to have a more complex sporophyte generation, right? All right, so this is kind of a review. Remember that plants kind of originally lived in the ocean, right? And their closest living relative is green algae, not sea anemones, as some of you put in the assignment. But yes, so green algae is the closest living relative of plants. And so plants originated in the oceans. But eventually the plants make it to land, right? And this isn't intuitive to you. Life in the ocean is way different than life on land, right? Life in the ocean, you can float around. You have water surrounding you, even though it may be slightly saline, but you still have water surrounding you. If anything, if you're underwater, gas exchange may be more difficult, right? So like think of life in the ocean. It's different than life on land. If you're gonna be successful on land, you have to have characteristics that are different than you would have had if you lived in the ocean, right? That makes sense. So plants show up on land around 400 million years ago. And this presents plants with a whole new opportunity because, like I said, land is way different than water. But in addition to that, the land is not homogeneous, right? The land is varied in itself. You have deserts, you have mountains, you have places where there's ice on the ground, places where it's hot all the time, right? So that presents additional opportunities for plants to move into these different habitats and environments. But in order to do that, they have to be able to live there. And so whoever is successful at living, for example, in the desert, will produce more offspring than things that are not successful at living in the desert. And over time, we'll see adaptation take place such that different plants become specialized to some of these different environments. So when plants first showed up on land, they basically did okay with having a dominant gametophyte generation. They did all right with that. Oops, I gotta move this. Right, as long as the gametophyte generation had certain features the plants did fine with the Gametophyte generation dominant, right? As long as they had a cuticle, as long as they had gamete producing structures and spore producing structures, that's the Gametangia sporangia there, that were multicellular and with a layer of protection with that sterile jacket. And as long as that zygote stayed inside of an archegonium and was somewhat protected, land plants such as mosses, liverworts, and hornworts did just fine with a dominant Gametophyte generation. But they need water for sperm to swim. So that alone is a limitation, right? If your Gametophyte generation is dominant, you're limited. You're limited in certain ways. You have to live in wet, humid places. Also, you don't have any vascular tissue, which is going to limit how many resources you could bring in and how much complexity and structure you can grow. So if plants are going to go into all of these different niches that are available on land, they have to evolve more complex structures. So they have to evolve structures that can acquire resources more efficiently, aka vascular tissue, also roots, stems, leaves. They need to evolve those structures. They need to be able to spread new individuals to further distances, because if you're just spreading individuals right where you are and you just came out of the ocean, then we will only have plants that are right by the ocean, right? And we wouldn't have any plants anywhere further away from the ocean. So if you're going to get to that mountain or you're going to get to that desert, you have to be able to spread new individuals to those locations, right? And lastly, if you're going to live in a place like a desert or a mountain, 3,000, 5,000 feet up, 20,000 feet up, or you're going to live in other areas that are just extreme, you need better protection for your egg and the zygote that led to that. So this, when you're reviewing this later, is a good time to go back to the pros and cons of being a non-vascular plant as being a vascular plant that I presented in lecture 4. All right. So what did we say? We need structures that can basically acquire resources more efficiently, meaning we need vasculature, roots, stems, and leaves. We need to spread progeny to further distances, and we need better protection for the egg. So in the bryophytes, mosses, liverworts, and hornworts, we have a really super small sporophyte plant that is completely dependent on the gametophyte plant. So if the gametophyte plant dies, the sporophyte plant dies, right? It can't live by itself. On the moss, here's an example of a true moss. That little brown thing coming out, that's the sporophyte plant. The green fluffy is the gametophyte plant. And the sporophyte is attached to the moss. On the liverwort, the sporophyte plants will be hanging upside down off of these archegoniaphores, right? But everything else here that we see, the thallus and the little palm tree archegoniaphore, those are all parts of the gametophyte plant, right? Sporophyte plant is just a little thing that hangs down off of this archegoniaphore. And when you get to the hornworts, the sporophyte plant is just this one horn. So very small, and most importantly, completely dependent on the gametophyte plant. The sporophyte plant cannot live on its own. Here's pictures of them in real life. You have true mosses on the left with polytrichum, and you have the peat mosses on the right, a sphagnum. Right, in true mosses, this whole little stick is the sporophyte plant. In peat mosses, this little olive looking thing is the sporophyte plant. Move on to the liverworts and hornworts. Same thing, right? The sporophyte plant is still, like I said, super small. Here, this yellow thing hanging down off of this archegoniaphore is a sporophyte plant in liverworts. And this horn thing, sporophyte plant in hornworts. So as we move through the plant kingdom, we get to the vascular plants, and now there's that switch, right? This is where that switch occurs. And part of the reason that switch occurs is because now the Sporophyte plant is, we need the Sporophyte plant to get more complex. And as the Sporophyte plant gets more complex, it ends up becoming the dominant generation. So check this out. I like to show this to students because sometimes it's not intuitive. You got on the top here non-vascular plants, you got on the bottom here vascular plants, important characteristics of both. Notice the dichotomy, right? Notice the opposing facts. Opposing facts. Non-vascular plants have no vascular tissue. Vascular plants have vascular tissue, right? Non-vascular plants have free-living gametophytes that are dominant. Vascular plants have free-living sporophytes that are dominant. And lastly, non-vascular plants have a sporophyte that is dependent and attached to the gametophyte plant. Whereas in the vascular plants, the gametophyte plant is going to become reduced, right? It's kind of like a bookend kind of thing. So when we started to learn about these about this vascular plants, right? Plants moving further onto land, having more complex sporophyte generation such that the sporophyte generation becomes dominant. And so if we're going to keep moving onto land, we have to have structures that can acquire resources more efficiently. So when we learned about the beginnings of these vascular plants, right? What were these structures that can acquire resources more efficiently? Well, in the bryophytes, but also in these kind of intermediate level plants, we don't have efficient means of transporting water and food throughout the plant body. We just have these cells called hydroids and leptoids that kind of work as pretty poor conductors, right? You can think of them almost as like leaky pipes, right? They're just not great at conducting things. However, once we get to the vascular plants, even the now extinct vascular plants that we can see just fossils, we get to xylem and phloem. So those are your structures that can acquire and move resources more efficiently. Increased complexity of sporophyte plants. So if you compare these here with sporophyte plants, you might notice the stems are smooth. There are sporangia. They're located at the tips of the plant, right, of the stems. But as we get, the sporophyte gets more complex, now we see a little differences. Now on this, we're seeing starting to see little like bumpy things on the sides of the stems. You might notice. Do you have a question? Okay. You might notice there's more complex branching going on here compared to like this. Here we only see this like very simple growth pattern, right? Oh, no problem, Brianna. I'm happy you're here. All right. Yeah, so here it's more of like a simple basic growth pattern. By the time we get to Rhiniophyta, you have more complexity. There's some pokey things on the sides of the stems showing up. And by the time you get to the Zosterophyllophyta here on the right, now, big leap in terms of sporophyte complexity. Now we have sporangia that are located on the sides of the plant, not just the tips. Sure, you still have some on the tips, but you have some on the sides now too, which increases your ability to make spores per plant individual because you can have more sporangia. We also see a new structure, which is these things called enations, that we're not exactly sure what they do, but it's still more complex. And we think what ultimately led to microfills, right? Right? We originally had these enations show up in the fossil record, which, based on what we know, didn't have any vascular tissue in the enations. They were just these little projections off the stems. But over evolutionary time, vascular tissue was able to enter those little projections, right, those enations, and lead to microfills, which were the first leaves, the very, very first leaves. So that's what this diagram here is showing. We went over that in that lecture. I think that's lecture four, or either 4 or 5. Yeah, so so microfills, those are the very first leaves. They are super simple. They have one vein only, and they have the tissues that we're used to in leaves, which is that epidermis, the mesophyll layer, which is where photosynthesis occurs, and then xylem and phloem in the veins, right? This is the type of leaves that Lycopodium and Selaginella have. They have microfills. In fact, this picture here, showing you a picture of Lycopodium, and below that drawing is from a slide through a cross-section of one of these microfills from Lycopodium. We learned about the different types of tissue. So notice the top here, right? We're still talking about the increased complexity of the sporophyte plant. So we went from sporangia at the tips and no leaves to sporangia on the sides and enation, then enation to microfills, right? Getting more and more complex as the sporophyte plant body. Now that the sporophyte plant body is able to have vasculature, right? Efficiently moving water and food throughout the plant body, we can have more complex tissues and more complex plant body parts like leaves, stems, and roots. And in the leaves, in the stems, in the roots, you'll have dermal tissue, vascular tissue, and ground tissue, which again, if you need to refresh, I think this is from lecture 4. And as the tissues get more complex, the way that the xylem and phloem are arranged inside of the plant also become more complex. When you start with the stele of Lycopodium, it's that simple, solid protostele. Once you get to Selaginella, you have the siphonostele with no leaf gaps. Once we get to the more later evolved plants, such as ferns, we'll see siphonostele with leaf gaps. And when we get to the seed plants, including the flowering plants, which is, again, 94% of all plants are flowering plants, we'll have this eustele shape of xylem and phloem arrangement. As the inside of the plant body gets more complex, such does the outside of the plant body. When we talked about Lycopodium and Selaginella, we talked about kind of these body plans of going from a very simple growing pattern like equal dichotomy, right? Let's use blue with a grow, split, equal, split, grow, split, equal, split. I forget who it was. Somebody wrote in the discussion board kind of having confusion over the idea of like equal dichotomy versus unequal dichotomy. And I did write an explanation there. If you still are confused, feel free to reach out to me and we could talk about it during office hours, because I want that to be clear. But grow and then split, equal, balanced, splitting. When you get to unequal dichotomy, you start to get unequal or unbalanced growth patterns, right? In that, like, grow, this one keeps growing. Maybe you have a split off this side, but this one not doing anything anymore, right? Whereas this one goes on to split again, then it sends up strobilus. So in Lycopodium, in the most simple ones, you have just equal dichotomy. In the ones that are a little bit more complex, you have unequal dichotomy. And by the time you get to Selaginella, you usually have a mix of the two. on plants, both equal and unequal dichotomy. Another way that the Sporophyte plant gets more complex is how many sporangia are able to be held on a plant individual. So I showed you in the previous slides that those very early vascular plants only had sporangia located at the tips. Later, we see the sporangia being able to be on the sides of the plant. In Lycopodium and Selaginella, sporangia is on leaves, which allows you to have way more sporangia than if it was just the tips. Also in Lycopodium and Selaginella, we see this new structure that I just mentioned before, strobilus. So this is actually two strobili that are on one stalk or one peduncle, but each strobilus contains leaves that are usually very reduced to be able to fit more of them in the strobilus. And on each of those leaves are sporangia. So that really increases your ability per plant individual to have to be able to make more spores. Likewise, when we get to horsetails, which we talked about last time, now we don't even have leaves that are bearing the sporangia anymore. Now we have these whole new types of strobiluses, of strobili, right, that have on them sporangiophores, and the sporangiophores that make these up. So like on this one here, for example, that's a strobilus, and on that strobilus are sporangiophores, and each of those sporangiophores can have like up to 20 sporangia. 20 is way more than 1, right? So the old strobilus, and this is what I drew last time, had leaves with 1 sporangia each. When you get here, you have strobilus composed of sporangiophores. Each sporangiophore has like 20 sporangia on it, way better than 1. So we needed the plant body to get more complex, meaning the sporophyte plant body. And we need it to be able to spread progeny to further distances. And this is kind of what I was talking about before, is that, like, you know, in order to spread progeny to further distances, the first step is to make more progeny. Like, if you only make 1 progeny, it can only spread however far that one spreads. If you can make 1,000 progeny, the chance of some of them getting further away is increased, right? Just by statistics. And this is just showing the slides of like what I just was saying, is that in the Lycopodium and Selaginella, there's only 1 sporangium for each leaf. And sometimes those leaves are not arranged in a strobilus, as shown in 1. Sometimes those leaves are arranged in a strobilus, as shown in 2. And if they are, then you usually are able to fit a lot more sporangia per plant, because there's so many leaves that are able to fit inside of those strobili. Yep. This is just talking about what I said before about growth pattern, going from more simple body to more complex body. When you get to horsetails, this is what I was just saying before, you now have strobili that are composed of sporangiophores, each sporangiophore being able to have many sporangia. So this here is 1 strobilus of a horsetail. Instead of leaves now, it has these things called sporangiophores on it. If we were to look, they kind of look like mushrooms. If you were to look at the sporangiophore up close, here is 1, right? Here's another sporangiophore. Underneath the sporangiophore, attached to the sporangiophore, are up to like 20 sporangia. Way more than 1 per leaf, but much better. So far in what we've learned in this course, we've seen a switch in dominant generation from Gametophyte to Sporophyte. We've seen increased complexity of the Sporophyte plant we've seen the ability to spread progeny further distances because now sporophyte plant individuals can make so many more spores than when we started this course looking at the bryophytes. Now what we need to get to the ultimate goal, which is a flower, we need better protection for the egg and for the zygote. And in order to get there, we have to go from plants that are homosporous to plants that are heterosporous, meaning sporophyte plants. Don't get confused here. So this top of the slide says size reduction of the gametophyte plant. And it says, in order to get to that ultimate goal, we have to make the egg on that gametophyte plant be better protected, and then the zygote too. But we can't get there unless the sporophyte plant is a heterosporous sporophyte plant. So this is Lycopodium. And so this is just to refresh you on that, what it is to be homosporous, what it is to be heterosporous. So if the sporophyte plant is homosporous, right, then the spores it makes are homosporous, meaning there's only one type of spore per plant, right? Here it is. And that spore will land somewhere, and out of the spore will grow the gametophyte plant. It will grow outside of the spore. My head is probably hiding it, but gametophytes grow outside of the spore wall is what it says here behind my head. And because there's only one type of spore, the gametophyte plants are usually bisexual, meaning have both male and female sex organs on them, aka monoecious, right? Remember, monoecious means one house. House is like a plant, one plant with both male, sorry, that's not male. One plant with both male and female sex organs on the same plant in one house, right? Is monoecious. So if the sporophyte plant is homosporous, it makes one type of spore, and the spore that it makes will grow into one type of gametophyte plant, usually a monoecious type that has both male and female sex organs on it. There's always exceptions. Who was homosporous that we looked at so far? All of the non-vascular plants, right? Here is the true moss and peat moss. Notice if your gametophyte plant grows outside of the spore wall, look how big it can be, right? These here are both gametophyte plant. True moss gametophyte plant on the left, peat moss gametophyte plant on the right. Relatively large gametophyte plants. As we move through the bryophytes, gametophyte plant starts to get a little bit smaller. In the liverworts, it was like a little green thallus. In the hornworts, an even smaller green thallus, right? But all these bryophytes have only one type of spore. Those sporophyte plants are only capable of making one type of spore. Once we get to Lycopodium, gametophyte plant is even smaller. Some species of Lycopodium make this little tiny green blob. This is not even comparable to a dime. This is more like the size of a peppercorn or something, super, super small. Other species of Lycopodium have a gametophyte plant that looks kind of like a radish or a turnip, also super, super small, that lives underground. But the point of these slides going through this is this size reduction of the gametophyte plant, right? Look how small the gametophyte plant is by the time we get to Lycopodium, which is still a plant that only produces one type of spore. But if we're going to get to that goal of a flower, we have to get to heterospory. The first time we see, and just an FYI, right, it doesn't go in a linear way, but horsetails also have homosporous sporophyte plants, right? So this is a horsetail gametophyte plant that is bearing both sex organs, growing outside of the spore wall, right? But if we're going to get to the flower, if we're going to eventually get to the flower, we have to get Heterosporous Sporophyte plants. And what do you know? Here we go. So once we get to Selaginella, that's what we have. That's our first time that's our first example in this course of encountering this idea of Heterosporous plants. On Heterosporous plants, I'll switch colors. If you're a Heterosporous Sporophyte plant, that means you can produce two types, or hetero, which means different, sporous, which means spores. So Sporophyte plants can produce two different types of spores. What are the types? You have microspores and you have megaspores. And likewise, you have sporangia that make one or the other, right? So some sporangia only make microspores. We call them microsporangia. Some sporangia only make megaspores. We call them megasporangia, right? Heterospory. If your spores are of two different types, I'll switch to orange. If your spores are of two different types, each type of spore grows to make its own gametophyte plant. If you are a megaspore, if you're a megaspore, you have inside of you a female gametophyte plant, which you can see here is trying, like just poking out just a little bit. And on that female gametophyte plant that's inside of that megaspore, there will be the sex organs. Inside of the microspore is the male gametophyte plant, but the male gametophyte plant has been reduced to a single antheridia, one antheridia. That's a huge reduction, right? If you think here on the top here, size reduction of the gametophyte plant, once we get to the microspore, think of a moss, right? So big, so green, so fluffy, whole organism, right? But by the time we get to microspores, inside the microspore is literally just an antheridium. Dr. Casper used to call microspores testicles in space, because it kind of is that. It's literally like the male gametophyte plant has been reduced to like a single testicle instead of a whole plant individual. That's one way you can think about it. My head is probably blocking it, but suffice it to say, if your spores are heterosporous, microspore, megaspore, and therefore the gametophyte plants are going to be, or at that point will be inside the spore wall, right? That's what I was saying here. The female gametophyte plant, she pokes out a little bit, but she's staying inside of this megaspore. And the male gametophyte plant, which is now just one antheridium, is inside that microspore. Okay, good. Thank you. Natasha says, your head doesn't block what you're showing us on our end. That's really good to know. Thank you. That will save time. Yeah, so gametophytes are, thank you. Right, Hawa says dioecious for heterosporous. Yes, but be clear with that, right? In that like it's the, here, is this sending to everyone? Hello. No, this is just sending to all right, everyone. Remember, right? It's homosporous and heterosporous. Refer to the sporophyte plant. Right? Homosporous and heterosporous refer to the sporophyte plant. But when you're saying here, yep. So it's okay to say like dioecious for heterosporous, that's okay, but I don't want you to get it twisted, because it's the sporophyte plant. Heterosporous or homosporous refers to spores. And what plant makes spores? Sporophyte plant. So you can describe the sporophyte plant as being homosporous or heterosporous. Dioecious and monoecious refer to having sex organs. Who has sex organs? The gametophyte plant has sex organs. So yes, if you have a, okay, good. yeah, if you have a heterosporous sporophyte plant, it will make two types of spores. They will grow into two types of gametophyte plants that will be dioecious. Very good. Okay, good. Yes. Let's move here. Right. So the whole point we're talking about this and going through this is because of the size reduction of the gametophyte plant. So if we look at microspores and megaspores under the microscope slide, microspores are super, super small, right? Each of them contains one antheridium. Megaspores are still very small, slightly bigger than the microspores. Each of those megaspores contains one female gametophyte plant. Okay. Why did we do this today? Well, because you guys have your first lecture exam on March 1st. So make sure you put that in your calendar. And as of right now, and it's February 17th, we have covered everything that's on lecture exam 1 as of today. So that means you can start studying for that exam because we've covered everything. What will the exam be like? The questions on the exam are going to be very similar in style and content to those assignment questions. They are not going to be identical, but they will be there will be a sense of familiarity, right? In that the content and style will be the same. After class today, I will post a study guide for lecture exam 1. So you will have a study guide for lecture exam 1 available to you 9 days before that first exam. More than a week. So I expect everyone to do kick ass on this first exam. I expect really good scores. I'm giving you a lot of time to, so if maybe you registered late for the course and you've been having a hard time catching up, this gives you ample time to catch up. And or maybe when you've done the assignments, you haven't really had time to review the material before you did the assignments, and then while you were doing the assignments, we're trying to look at the PowerPoint slides and look at your notes and getting all confused and whoops, ran out of time. Now you can take time to really study for this exam because I want you guys to do well. No, Hawa says, can we have a little more time for the, oh, do you mean Hawa, do you mean like the actual exam itself? Meaning, okay. Well, obviously the exam will be more time. Yeah, obviously the exam will be more time than the times that the assignments are, right? Because the exam will have more questions. The exam will be something like, I haven't written it yet, but there'll be something like 40-ish questions that will be similar in style and content to those we've seen in the assignments. I'll give you ample time. Let's put it this way. If you're prepared, you most of the time, yeah, most of the time when I give these exams, the amount of time that I allot for the exam, I'd say 80 to 90% of the students use maybe half of that time to maybe 75% of that time. And there's usually about 20 to 25 students that use the entire time. Some of the students who use the entire time are students who like me, in that even though I already answered all the questions, I want to torture myself and look at all the questions again and again and again until time expires so that I can make sure I did everything right, which is a strategy I don't recommend, but that's a strategy that I did during, so I understand if you're that person. With the rest of the time today, what I want to talk about is studying, because based on some of your assignments and stuff, it's something we should talk about. And this is also something I think undergrad programs do a really bad job at dealing with. So like when I was in undergrad, I was frustrated by the fact that basically I would go to class, I would be shown material to learn, and And then I would be expected to kind of know how to take that material, smash it into my brain, and then be able to hold on to it during an exam, which is already a stressful situation, right? And I felt like undergrad didn't do a good job of kind of telling me strategies of how to study. It was kind of like assumed like, oh, you're in college, so you must know how to study. But that's like not the case. Just because you're in college and learning hard material does not mean you know how to study. And in fact, I had to do a lot of trial and error to figure out what worked for me in order for me to be able to retain the information. And as an undergrad, most of you are fourth year students, so hopefully you've gotten a sense of what works for you and what doesn't work for you at this point. But if you haven't, there's still time to kind of hone that in. If anything, I would say, if you're doing something that requires a lot of effort and you're not getting the results you want, you might want to switch strategies, right? Like if you feel in your heart, like I'm putting in the time, I'm studying this material, but I'm not doing well, you might need to switch your tactic. So I'll talk to you now about a little bit about what works for me. And this may not work for you, but at least it's somebody telling you what works for them. And for those of you who have plans of going to higher education after this, or even for those of you who plan on going directly into work and you may have to do a presentation or talk to colleagues about a complicated topic, this is something you could do to maybe hold on to the material better. So before I do this, I want to say, how do you study for exams? Like, this is what I picture, like, because I've walked around campus in the before times and I've seen students studying for exams. And what I see, and what I myself used to do in my beginning part of undergrad, is you basically sit and you reread. Oh, it should say your notes, not you notes. Do you sit and reread your notes? No, it should say your notes. But like, do you do that? Do you literally like, okay, everyone, leave me alone. I'm turning my phone to silent. I'm gonna go study now. And if you're not turning your phone to silent while you're studying, that's, yeah, you should do that too. Phones can be very distracting when you're trying to study. But do you literally like, okay, here, I got the book, I got my notes. Maybe I have some diagrams that are like already filled in with the correct answers. And I'm just gonna literally like sit and stare at them for a few hours. We got a raised hand, I think. Do we have a question? I thought I saw somebody have a question.
Oh no, I didn't know that was a rhetorical question. I thought you were genuinely asking us.
Well, okay, so hey, but all right, so Aya, how do you study for an exam?
So usually, I have I distinguish between my notes. So I have like the notes that I either get from a textbook if like the professor says like the textbook is a good resource to use, and then also the lecture notes. But like for this class particularly, I have like the slides where you have like those rigid like points that you write out, but then when you're talking, you obviously elaborate on those points. So I watch those recordings and then I like add in notes of my own based off of what you said. And then after that, that's what I do in school. Like I'll spend time writing them down and like making like really extensive notes. And then when I go home, I like to talk myself through the concepts. Like I like to pretend like I'm teaching somebody else. So like I'll talk myself through it and then make it make sense to me.
Yeah.
And then if there's like necessary like diagrams or stuff, I try to draw them, or like I'll just pull them up on my computer and like be like, this thingy looks like I try to relate it to some sort of like, I guess like put it in like more layman terms just because it's easier to remember.
Sure.
But yeah.
Sure. And so that kind of is a perfect segue into leading where I was going to go with this, in that these things that I have written here, sitting and rereading the notes or staring at diagrams that have already been filled in with the correct answers. The reasons why these typically don't work is because you're not really engaging the content that you're trying to learn. And what I was just explaining is that, you know, by talking yourself through it or redrawing diagrams or, you know, putting these complex things into more layman's terms. All three of those are examples of her engaging with the content. And that type of engagement is what makes things stick better. So thank you for sharing your strategy with us. Oh, take-home message, don't do this. Don't sit and reread your notes. Don't stare at diagrams that are already filled in. That doesn't work. At least it didn't work for me. Maybe it works for you, but it doesn't work. Let's use an example. Let's make collage. Maybe you've heard of collage before. Maybe you haven't heard of collage before. Collage is something that my grandma used to make. I think it's delicious. But basically, let's make collage, right? Okay, here you go. Here's the instructions. Here's the ingredients. Let's say you stare at this. I tell you, okay, you're going to make me collage later today, right? So I give you this, and you go and you sit and you stare at this for some time, right? 10 minutes, 20 minutes, and you just stare at it, right? Then I take it away from you. I take away the instructions, I take away the ingredients, and I say, all right, go on your way and make me collage, right? Go to the store, buy all the right ingredients, bring the ingredients back, make the collage with no help, no instructions. Probably not going to be able to make it, right? Because reading and staring are not good ways to get the material to stick. If you, on the other hand, gave this to your friend and said, okay, all right, I just stared at this for a while. So, okay, friend, let's see. First thing we need is some flour. Oops, nope, wrong crap, right? So this like kind of trying to recall is a good way, All right. So what I recommend is, first of all, if you can, study in pairs or small groups. I know you're super busy. When I was in undergrad, I worked at least 30 hours a week, if not more. I was a full-time student, and I did a bunch of different extracurricular activities on campus, and I had a life also outside of school. So I get that it can be really difficult to find time to work with your colleagues, and I get that also, well, now last semester we were fully virtual, so it was really hard for students to meet one another. Now you're in lab together, so you can at least maybe reach out to somebody who sits next to you in lab and be like, hey, can I give you my email address? Maybe can we try to study together at some point? You don't have to do this in person. You can do this virtually. You can do this via Zoom. You can do this via Duo. You can do this via FaceTime. You can do it via all those things. I see people on their FaceTime or their video chat, whatever platform they're using, all day, especially students, right? Hi, yeah, I'm walking. Yeah, oh yeah, this is, look, here I am walking, right? Pepper some of that in with, hi, yeah, so bryophytes have Gametophyte generation dominant, right? Pepper some of that in. Yeah, it really does help because it makes you accountable. Whoops. But maybe you're busy, maybe you don't have time to do that. And so if you don't have time to do that, or you're shy, which is also okay, I have developed this kind of quiz yourself method that works for old school ways of like printing out paper and new school ways, meaning saving a PDF and it's on your phone. So what does it look like? This is how you do it. This might seem like a no-brainer, but believe it or not, this is, a lot of people maybe didn't think of doing this. So these are what I call updated flashcards. What you do is you open a Word document and you change the orientation to landscape. And then you insert a table that has 2 columns and lots and lots and lots and lots and lots of rows. On the column on the left, you put questions or unlabeled diagrams or pictures you took in the lab or terms that you want to, that need to be defined. And you put that in the left-hand column. And what's nice about this class is you have the lectures as PowerPoints. You have the lab manuals you have the lectures as PDF slides, right? You have the lab manuals as PDFs. So you can easily take screenshots of lab content or whatever to get those unlabeled diagrams into this flashcard system. On the right-hand side in the columns, you put the answers or a labeled version of the diagram or the definition to the term. Then you have the option, if you like to work with paper, you could print this out and fold it in half. And then you can be only looking at the questions, and then you can be like, okay, I think that is pointing to the xylem. All right, let me look. Nope, it wasn't. It was pointing to the epidermis, right? Okay, got to redo that one. Or what I like to do, because I used to commute like 3 to 4 hours a day on the subway, and I'm sorry, having regular flashcards or having a freaking piece of paper to fold during rush hour, sometimes not so easy. So what I like to do is I print this, save this as a PDF, email it to myself, and then this way I can look at it on my phone. And what I could do is I could zoom in to see only the questions, then have that recall movement of, okay, I think it's this. Let me look at the answer. Okay, I was right, right? Here's what it might look like. This is just example, right? So like you could like zoom in. If you had this as a PDF, you could like just be zoomed in to this side. What does monoecious mean? And then you would have to recall, it means a plant that makes money. Let me look. No, it's not. Okay, crap. I need to study that more, right? Let's see. If you build up the left column of questions and don't have answers, should you look them up or should you go through the slides? I don't really understand what you mean, Henry. If you build up the left column of questions but don't have the answers, what I would recommend.
Like, let's say
go ahead.
you're setting up all your questions to study from, but you just don't have the answers yourself. So like, do you just? I mean, like it's like this. Like, let's say you put up on the side, what does dioecious mean? And
Yeah.
you've seen the word, but you don't remember. I know what it means, but you've
Yeah.
seen the word. What would you do? Would you just like look it
Right.
up on Google, or is it better to look at just the slides?
Right.
Because to
So
me, I feel like that takes
definitely.
too long, but it
Right.
can be the best answer, you know?
Right. Well, so that's a good point. Right. So your best resource for finding the answers to these questions is the lecture slides, followed by the book, followed by lab manual content and your notes from lab, followed by the internet. Internet use with discretion. Remember, Google is not writing your exam. I'm writing your exam. Google is not teaching this course. I'm teaching this course. So when I tell you what monoecious means, that's what I want you to know about monoecious. If Google tells you monoecious is a Rihanna single from 1992 that was never released or something, right? That's not what I want. And that's not true. But I'm just saying Google will give you an answer that Google thinks is correct based on whatever algorithms have told Google it's the most popular response. But your best resources here are the lecture slides, your notes, the book, the lab materials. If though you have a question that you just cannot for the life of you find the answer to, reach out to me. I can help you find the answer. If I think it's an obvious answer, I might start with just pointing you back to a specific lecture or even a specific group of slides from a lecture. If it's a more complicated answer, I'll just tell it to you. I'll just tell it to you. So reach out to me too. Or, even better, reach out to me or write the question in the discussion board. And then maybe one of your fellow students could answer it for you, or I'll jump in there and answer it. Okay. The reason why I'm doing this is because these are your current assignment grades. So I finished, as of right now, on Canvas. Everything's been graded that you turned in so far, which is just these 2 assignments. And I've calculated attendance and participation points as of yesterday, but that does not include lab attendance yet. I'm going to get that from Dr. Fonseca. And it does not include attendance from the lecture 8 or discussion board for lecture 8 yet. Just keep that in mind. And so far, I'd say most of you are doing okay, but I'm getting a little bit of a red flag. I don't know if you watch Catfish and like Candy with her red flag. I'm getting some red flags here because of a trend I'm looking at right here, right? So if you look at assignment 1 grades, most students got A's, right? 16 out of 39 students got A's, followed by B's, followed by C's, couple D's, and most happily, 0 F's for that assignment 1, right? Which is great. However, once we get to assignment 2, holy cow, what is happening? What is happening? This is not good. We got a bunch, so now we only have 7 A's. Many people have slid down this, and oof, 11 F's on assignment 2. Now, some of that is due to, there's a few of those F's that were due to late submissions, and 4 of these F's are because students didn't even turn in the assignment. Not an excuse, not good. You have a week, each assignment, there's only been 2. You have a whole week to do it. And it takes about 20 minutes to do it, give or take. And I put reminders on Canvas, and I say reminders in lecture for these assignments. So there is really legit no excuse to not turn in the assignments. You really have to treat yourself better and make sure you do these assignments. Remember, all assignments collectively count for more points in the course than a single exam. Maybe you're looking at this and saying, well, you know what, Rebecca, this class is hard, and also it's virtual lecture, so you know, I can't, no, don't even, because this is the same types of assignments from spring 2021 when I taught this course. And this course was completely virtual, including lab in spring 2021. And look at my assignment 1 grades. I had 23 students with A's, and my assignment 2 grades, I had 11 A's. You can see the trend. Yes, there were more F's. Again, that was because of students not turning stuff in. But yeah, this is why I'm mentioning this now. You have an assignment due Thursday, or no, you had an assignment due this morning. Yes, hopefully you turned it in. If you didn't, you still have time to turn it in, and you can still get points for that. And yeah, so the reason why I'm showing this to you guys is because I've taught, this is my 5th time lecturing this course. A student's average assignment grade usually predicts the student's exam performance grade. So like if a student is averaging like an 80 in assignment grades, they'll usually score between like a 75 and an 85 on the exam. So that's why I'm mentioning this, because I want you guys to do well in this course. All right, the first exam is on March 1st. It's 9 days from now. There will be a study guide available by 11:30. So in like 10 minutes, sorry, 20 minutes. And yeah, what I recommend, download the study guide, use the study guide. If you're gonna use this updated flashcard system, use the study guide to make the questions in that updated flashcard system. Yeah, but you have 9 days to study. I'm here if you need me, reach out. In the meantime, we have a little bit of time now. So does anyone have any questions about anything, or need clarification, or confused about anything? Or maybe you got something wrong on an assignment and you're not sure why. I'm happy to talk about that now, or you can send me an email to talk about it. Either way. One thing I should mention, maybe while you're thinking about questions, is how is attendance and participation graded, right? If you attend lecture, you get a point for attending lecture. If you are more than 10 minutes late to lecture, you start to lose parts of that point, right? If you show up to lecture, or if you leave lecture early, right, you start to lose points as well. But if you are in lecture the whole time, you get 1 point. So that's the attendance component. Participation component is the discussion board threads. In the beginning of the semester, I wasn't clear about the timing. So those first several discussion board threads, it's basically like, I don't take off points if you send it late. But I made an announcement, you can see it on Canvas, I think around the 24th or so, about how the participation points will be dealt with in the discussion boards. So after a lecture is over, you should, within 2 days, post to that discussion board. If you post after 2 days, you get half participation points. If you post more than a week, but it could even be up to a month after the lecture, you get quarter points for participation. If you don't post anything at all, you get 0, right? So for some of you who have not been active on those discussion boards, you can still go back and put content there that you will get some points for. So I encourage you to do that.
We can just comment on somebody else's something for points, right?
Yep, that counts. Yep, that totally counts. I have it set up so you guys can write on each other's threads. Yep, that's totally fine. And again, it doesn't have to be anything profound. It can just be a simple comment or like, I liked this class. It's just for participation, right? You might say, well, I think that's kind of unreasonable for you to like give us timelines to post on the discussion. Not really though, because the whole point of the discussion board is for me to be able to address questions in real time as we go through the course. So like right now, if you post something on the Moss one, yeah, I mean, I can answer your question, but if that was a question that you had way back during the Moss one, it's to your benefit that I answer that while we're actually going through the material, as versus like once we're way, way far away. That's why I give you the timelines. Okay. If no one has questions or comments or concerns, we can end for the day. Yeah, so I hope you guys have a great day. I hope you don't mind me nudging you a little bit, but I want everyone in the class to do well. I want you all to graduate. I want you all to move on to other things. And I know it's hard to juggle everything that you have going on, but some of you need to put more time into this course. Oh, speaking of which, right? Last Thursday is a pre-recorded lecture. Last Thursday, right? Tuesday night, I look at YouTube and only 22 people had watched, and I'm including myself and my other people who I know who look, but as of Tuesday night, only 22 views that it couldn't be accounted for. And I have 39 students in my course. What kind of math is that? And so that was a Thursday morning pre-recorded, Tuesday evening, still like 17 people at least had not watched the Thursday recording. That is just makes me want to cry. I can't help you at that point. Like if you're not gonna be watching it, there's nothing I can do for you. It's just, that's it. But so yeah, if you're one of those people, please go watch that. Please go watch that lecture. All right, enough. I just want you guys to do well. I care. All right, bye everyone. Have a good one. I see a message here asking to speak with me privately. Could you send me an email and I'll send you a different link for us to do that. Thank you. Have a good one, guys.